In Chapter 8, we focus on the chemistry of DNA, so the structure of the individual nucleotides that make up our DNA molecule. And we learn how the structure of our nucleotides affects and produces the overall three-dimensional structure of our DNA molecule. Also included in this chapter is a little bit of history about how we came to understand the structure of DNA and DNA as a coding molecule. And then we'll finish up with a little bit about chromosome structure and genome structure. So how did we get to the structure of DNA? We didn't fully understand the structure of the DNA until the 1950s. In 1953, Watson and Crick published their paper outlining their determination of the structure of DNA. But their understanding of the structure was based on many years of individuals learning about the structure, the chemical structure, and also the nature of it as a genetic coding molecule. And once we got this structure, once we understood the nucleic acid structure and the overall physical structure, we could then begin to understand how DNA replicates, how DNA is expressed, which we now know as transcription and translation, and how DNA works for a coding molecule. Our genetic material has to possess several key characteristics. The main function is that it has to contain complex information. If we think about what our genetic material does, it stores information and then that information is passed on to our next generation, whether that is the next generation of cells or the next generation of organisms. So it has to store information and it has to be able to pass on that information. But it also has to use that information to build and run a living organism. And so not only does our genetic information have to, our genetic material have to hold that information, it must be able to replicate it to pass on either through mitosis or meiosis and reproduction, or through the process of binary fission, which is how our bacteria replicate. Our genetic material must also be able to encode the phenotype or the physical characteristic of an organism. But these phenotypes are not always just the physical outside manifestation of our genes. It's also all of the metabolic processes that run the organism. So it's what we see, the physical characteristics, but it's also how we work and how we run. And all of those instructions have to be encoded in the DNA as well. And then finally, genetic material must have the capacity to vary. This is going to be the basis of our understanding of evolution and change over time. Because our most basic definition of evolution is the change in the genetic makeup of a population. And we also know that genetic material is not identical from parent to offspring, except in the case of asexual reproduction. But if we're doing sexual reproduction, our parents and our offspring all have different DNA. Both parents have different DNA, and then the offspring are combinations of those parents. Sometimes new combinations that we haven't seen in other siblings. And so there has to be mechanisms within the genetic coding system that is going to allow the system to vary. And that can either be by recombination, which we have seen in meiosis, but it can also be through mutation and accumulated changes in the nucleotide sequence of our molecule. So why was the discovery of the structure of DNA so important? Well, without an understanding of the structure, we couldn't figure out how DNA was really working. And we see this common theme in biology that structure is related to function. And the specific structure of an organism is also going to give it its specific structure of a molecule in this case is also going to give it its particular function and its ability to work. So the structure of DNA has to reflect its ability to store information, to replicate information, to express that information, and to change that information. And without the structural support for all of those processes, our genetic material wouldn't function in the way that we need it to. And so we needed to understand that chemical structure in order to understand how we get phenotypes, how we replicate our DNA. Cell biologists, also called cytologists, and chemists have been working on the structure of DNA and function of DNA for quite a long time. As far back as the mid to late 1800s, individuals were looking at these chromosome structures that were found inside the nucleus of the cell. In the late 1800s, Johann Miescher isolated chromatin from the nucleus of a white blood cell for the first time. Chromatin is a structure that consists of both the DNA molecule and proteins. And we'll talk about what chromatin is a little bit later in our lecture for this chapter. But chromatin is a complex made up of DNA and proteins. Originally, these structures were called chromatin and chromosomes because the word chroma means color. And when individuals were first looking at these under the microscope, they were staining cells to be able to see them better. Because if you look at an untreated cell under a microscope, it's mostly going to look clear. And so in order to see contrast between structures and to see the different structures, scientists were staining cells, and they were using different types of dyes to stain different parts of cells. And so the word chromatin comes from the word color. These are colored structures that they were seeing in the nucleus. Also in the late 1800s, Albrecht Kossel determined that DNA contains 4 different nitrogenous bases. And then chemists continued to work on this, and Phoebus Levene, who was working in the early 1900s, identified the components of those 4 nitrogenous bases. So he discovered ribose and deoxyribose, and then put that together with Kossel's understanding of there being 4 different nitrogenous bases. Levene proposed the tetranucleotide hypothesis, which states that DNA is made up of 4 different nucleotides. And of course, now we know the 3 components of the nucleotide, the phosphate, the sugar, and the nitrogenous base. And our nucleotides vary by that, I'm just going to abbreviate it, nitrogenous base. As we go through the rest of this video, we are going to look at some of the history and experiments that led to our understanding of DNA structure and molecular function. There are 5 or 6 experiments or groups of people who contributed important pieces of understanding to this puzzle. On your next exam, you will need to know each of the individuals, the experiments that they did, what they found, and why this is important. And then there is one other experiment in chapter 9 that you'll need to know that is related to DNA replication. So we're going to start off with Erwin Chargaff. He published a paper in 1950. So Watson and Crick published their structure information in 1953. So there were a lot of people that were working on the structure of DNA at this time. Erwin Chargaff was a biochemist who was looking at the composition of genomes. And he was looking at the concentration of each nucleotide within the genome. So he wasn't doing any kind of sequencing. He was doing analysis to determine how much of the total weight of the DNA molecule was adenine, thymine, guanine, and cytosine. And he looked at a variety of different organisms, determined the base composition, the percentage of bases in each of these organisms. And he noticed a trend. He noticed that the the percentage of adenine by weight was always the same as the percentage of thymine by weight. And so if we look at E. coli here, we've got 26% of our sample of DNA is adenine and 23.9% is thymine. So these are relatively close. And then he noticed that for guanine, 24.9%, 25% for cytosine. So he recognized that in each species, although the actual percentages varied, the percent of adenine was always similar to the percent of thymine, and the percent of guanine was always similar to the percent of cytosine. So no matter what organism he looked at, whether it was a bacteria or a eukaryote, he found the same data. So if we look at humans, we see this is even, our values are even closer, 30.3, 30.3, and then 19.5 and 19.9. And he continued to see this no matter what he did. The columns on the right show the ratios of adenine to guanine, showing that, again, that they are roughly equal. So in 1955, he published a book that outlined all of his experiments. So if we look down here at the bottom, it actually says 1955 rather than 1950. But that initial paper was published just a couple of years before Watson and Crick published their paper on DNA structure. So Erwin Chargaff, his data was very, very helpful in Watson and Crick's ability to be able to produce the overall structure of the DNA molecule. So around the same time that chemists, biochemists, and biologists were working on the structure of DNA, other researchers were working on essentially how DNA functions, how our molecules in our cell function as genetic coding molecules. And so we have 3 experiments that show us that DNA is the molecule that encodes our genetic information. Besides DNA, the other candidates are RNA and protein. Mainly protein. And the reason that we say mainly protein is because remember that chromatin is made up of both DNA and protein. And the function of RNA wasn't really figured out yet, but the 2 candidates for encoding that genetic information were DNA and protein. But it was actually generally thought that protein was going to be that coding molecule because protein structure is much more complex than DNA structure. And so the idea here was that if we have a super complicated genetic coding molecule, then it's probably got a super complicated structure. And protein has the ability to be far more complicated. So we're going to look at 3 experiments that led to the identification as DNA as the transforming principle. And we'll talk about why we're calling it the transforming principle right now. Eventually, we're going to start calling it the coding molecule because we're going to learn how it encodes our genes. So you'll need to know Erwin Chargaff's contribution. You'll also need to know about Griffith's experiment, the Avery, McLeod, and McCarty experiment, and then the Hershey and Chase experiment. Not listed here are going to be the contributions of Watson and Crick, Maurice Wilkins, and Rosalind Franklin. For Griffith's experiment, I'd like you to think about the process of transformation. We talked about transformation in chapter 7 as one of the ways that bacteria share genetic information. And remember that we said that transformation is the uptake of DNA from the environment. And that is the basis of Griffith's experiment. So we're going to go through the setup for Griffith's experiment, then we're going to talk about what happens in this experiment and how it tells us that DNA is that transforming principle. So to start off, we are using disease-causing bacteria, specifically Diplococcus pneumoniae, which causes pneumonia. Griffith was using mice, so he was essentially exposing mice to pneumonia-causing bacteria and then watching what happened to them. Mostly, did they live or did they die? And he wanted to figure out basically the mechanism for this viral transmission, but he wanted to figure out if there was some sort of principle that caused the mice to die. Was it the whole cell? Was it some particular part of the cell? So Griffith inoculated these lab mice with 4 different types of Diplococcus pneumoniae. The first was the virulent bacteria, so the 2S virulent bacteria. These bacteria are going to be disease causing. And so I'd like you to think about what this is going to do to the mice. So if it causes disease, it's probably going to kill them. Next was a non-virulent strain, so a strain of Diplococcus pneumoniae that was not disease causing. So not disease causing. Then we've got heat-killed virulent bacteria. So if the idea here was if we kill the cell, then the organism should not be infected. So there should be no infection, even though he has the disease-causing strain. Because they've been killed, so he's trying to determine if the heat-killed bacteria, even though they have the information for causing virulence, whether they do or not. And then he mixed 2 strains together. He mixed the non-virulent with the heat-killed because he was really trying to figure out what component was it that was causing this virulence in these bacteria. So on the right-hand side here, we've got a nice little depiction summary of the experiment. I would suggest, because it's small, that you look at it in your textbook. It should be in section 1 or 2 in chapter 8. So summary of the experiment. Not surprising, those virulent bacteria killed the mice that they were infecting. The non-virulent survived. Then he looked at those heat-killed virulent bacteria and he noticed that the mice survived. So this indicates that there's something about the live bacteria that's causing the infection. But then things got interesting when he looked at that mix of non-virulent and virulent. So non-virulent by itself shouldn't cause any problems. Those mice should survive. And he had showed that the heat-killed strain didn't kill the mice either. But what he did see when he mixed them together was that the mice with both of these strains, infected with both of these strains, died. So what happened? Why did these 2 strains that didn't kill the mice kill them when they got together? So Griffith concluded that there was some sort of factor, some sort of transforming principle that was transferred from those heat-killed strains to the non-virulent strain. So this meant that there was a factor separate from the living cell, or at least able to be separated from the living cell, that was transferred to another cell. So this is where we get the name transformation. And the idea here is that those heat-killed cells must have released their DNA, and that non-virulent strand must have picked them up, because we need a living cell in order for the mice to contract pneumonia. And so that transforming principle, or it turned out that there was a transforming principle that could be transferred from one bacteria to another. So Griffith essentially showed that there was some factor, and we don't know what it is yet, but there was some factor that could be transferred along with its ability to kill these mice between bacteria. Now we know that in the process of transformation, cells pick up DNA from the environment around us. Remember, this is chapter 7. So the cells can pick up that DNA, incorporate it into their cells. So what's really happening here, now that we have all of the details, what's happening here is that the DNA from that heat-killed strain is being transferred to the non-virulent through the process of transformation. So to continue with this idea of transformation, another research group, Avery, McLeod, and McCarty, wanted to determine what that transforming principle was. And so they designed this experiment to essentially isolate the top candidates and see which one by itself could be that transforming principle. So there were 3 options here. They wanted to test to see if that transforming principle was RNA, protein, or DNA. So they took that virulent bacteria, that disease-causing strain, and separated it into 3 groups. To one of those groups, they added RNase. RNase is an enzyme that breaks down RNA in the cell. So from Biology 101, we know that an ACE is an enzyme. It's kind of the general term for an enzyme. Anything that has ACE on the end is an enzyme. So RNase is an RNA enzyme that cuts up RNA. So they added RNase to one sample of these virulent bacteria, and then they inoculated mice. To another sample of the bacteria, they added protease, which is an enzyme that breaks down protein. And then to a third group, they added DNase, like a restriction enzyme that inactivates DNA. It does this by cutting up DNA. So RNase breaks down RNA, protease breaks down protein, DNase breaks down DNA. And so they wanted to see what would happen when they inoculated mice with each of these treated samples. So the first thing they did was inoculate the bacteria with the RNase treated cells, and that caused the mice to die. So transformation still occurred in the cell. RNase did not stop that transformation. So this means it was not RNA. That transforming principle was not RNA. When they treated the sample with protease to break down the protein, again, the mice died. So protein was not the transforming principle. So now they know that it is not protein and it's not RNA. When they treated the cells with DNase to break down the DNA, the mice survived. This suggests that DNA was the transforming principle. So what does this mean besides the fact that we can kill bacteria with this transforming principle? Well, what it means is that DNA is what is being passed from cell to cell. Now, in the bacteria, this is happening through transformation, which is why we call it the transforming principle, because we saw that transformation happening in Griffith's experiment. But this could also suggest that DNA, because there's some sort of coding factor in that DNA that is acting to kill these bacteria, to kill these mice, it could mean that DNA is also the inherited transforming principle, how genetic information is passed from parent to offspring. Our next experiment is similar to Avery, McLeod, and McCarty's experiment, except that it uses bacteriophage. In this experiment, Hershey and Chase demonstrated that DNA is the genetic material that bacteriophages use to infect their cell. So this is along the same lines of Griffith and Avery, McLeod, and McCarty demonstrating that DNA is the transforming principle from bacteria. So they wanted to figure out what the infectious particle was in our E. coli virus, that bacteriophage. So remember that bacteriophage have this protein capsule at the top with genetic material in it, and then they've got this neck, and then they've got legs that attach to the cell. Now, we learned about viruses in a previous chapter, and we learned that viruses inject their DNA into the cell. So we know that now, but in the 1950s, researchers wanted to figure this out. So they started by growing viruses in two different cultures. In one of those cultures, they added radioactive sulfur. And the reason that they grew these bacteriophages in the presence of radioactive sulfur was that sulfur is found in proteins, but not DNA. And so they could essentially label their proteins with this radioactive sulfur. And then they grew another round of viruses in radioactive phosphorus. And this is because there's phosphorus in our nucleotides, but not in our proteins. So they ended up with viruses that are labeled with radioactive phosphorus. So then once they had these two populations, the two populations of bacteriophages that were labeled individually, they decided to let these phages infect the cell. So what we get, two different cells, two different solutions, one with that radioactive sulfur for the proteins, and I'm going to make our DNA, nope, we're not doing green because green's already there. I'm going to make the DNA purple for now because we know that that's separate. But so we had these tagged proteins, and then in the other group, we had our untagged protein, but now we have our tagged nucleotides. So one set of viruses should infect, if it's proteins, with radioactive protein. The other group would infect, if it's nucleotides, with this radioactive phosphorus. So they let the viruses attach, let them sort of incubate together just long enough to let viruses start to attach, but not long enough for these cells to start to burst. And then they put them in a blender and basically knocked off the viruses from the cells, and then put them in a centrifuge and separated the cell debris from the solution that contained the virus particles. And so what they would have expected is that if the cells contained the sulfur, they would be down in the bottom of their tube. And if the cells contained DNA, then they would have cells with phosphorus down in the bottom. And then the solution up in the top would have the remnants of the infection. So they took the solution off and then analyzed those cells to figure out which ones were infected by this infectious particle. It turns out that it was the cells that were infected with phosphorus. Let me restate that. It turns out that the cells infected with phosphorus were actually the infected cells, the ones that were capable of producing new viruses. So they recognized that bacteriophages were transferring DNA because when allowed to infect the cells, DNA was what was transferred and therefore what was radioactive in the cells. It was not the proteins, because the radioactive material remained in solution, not the cells. So the radioactive proteins were not transferred into those cells. So here we have a summary of the results. After they put their cells into the blender, centrifuged it off, they found the radioactive phosphorus inside the cells, but no radioactive sulfur. So just like the Avery, McLeod, and McCarty experiment, they determined that DNA was the not transforming principle, but infectious particle that could cause disease. In this case, it is new viruses. So now we get to the early 50s. We've got Chargaff explaining his equal amounts of adenine, thymine, and guanine and cytosine. And we've got this understanding that transforming principle in bacteria is DNA, infectious particle in viruses is DNA. And so now we have an understanding of what molecule is actually acting as our coding molecule. So we now know that it's DNA. We know that DNA is found in all of our cells. And so Watson and Crick were interested in determining the overall structure. There were quite a few chemists, mostly chemists, but quite a few chemists, biochemists, biologists who were interested in determining the structure of DNA. Watson and Crick were actually not working on the structure of DNA formally. They were doing it as a side job, something that they were kind of interested in. They actually had been told they shouldn't be working on it. They should be doing their primary research, but they did not listen. Also working on the structure of DNA was Rosalind Franklin. So Rosalind Franklin was at a separate university. She was working begrudgingly with another individual named Maurice Wilkins. So they were both taking X-ray diffraction images of DNA molecule, but Rosalind Franklin was doing most of the work here. So essentially what she was doing was shining an X-ray on crystallized DNA. And when you shine light, X-ray, whatever onto something, it bounces back and it produces a particular diffraction pattern. So anytime that light hits something, it bounces off, and it bounces off at a particular angle. So Rosalind Franklin was using X-rays. Basically, X-rays travel at very, very short wavelengths. You need short wavelengths for tiny molecules. So she was blasting tiny molecules with X-rays and exposing their diffraction pattern onto film. And she had a variety of pictures. Her famous one is called Sp26, and this is B-form DNA. We'll talk about what that means in a few minutes. And then we've got another picture over here. This is the A-form DNA. And you can see they look a little bit different. And if you were well versed in translating X-ray diffraction patterns, you would understand what this was saying. So Rosalind Franklin was taking these pictures. She was analyzing these pictures, looking for the overall shape of DNA. So she was doing X-rays. Watson and Crick wanted to build their molecule. They wanted to look at the physical structure, put in the nucleotides, and kind of check how they were all arranged. But the leading theory at the time was actually that DNA was a triple helix. And there were some famous chemists that were trying to build this triple helix, but none of them could really get it to work. They had to sort of make up some chemistry rules to make it work, and that probably wasn't really going to work in a real cell. So Watson and Crick were working separately from Franklin, who was working separately from Wilkins, but they were both working in the same lab. So Watson and Crick didn't really like Rosalind Franklin. Rosalind Franklin didn't really like anybody, and they kind of became buddies with Maurice Wilkins. Well, Maurice Wilkins showed them some of Rosalind Franklin's work, including photo 51 from the previous page. So they looked at that photo and they recognized that the X shape in the center, so it's kind of shaped like this, and then a little dot in the middle. They recognized that that X shape indicated a helix. And then based on the pattern of these dots, recognized that it meant it was actually a double helix. And then they could also use the distances between these different shapes to determine the width and the turn ratio of that double helix. So they took this information and all the previously known information, like Chargaff's information about the chemistry of the nucleotides, because remember, we've already figured out the nucleotides and what they're made out of. So they knew the structure of the individual nucleotides, and then based on this idea of the double helix and the complementary base pairing, they were able to build this large man-sized ball and stick model of DNA. And then very shortly after that, in 1953, they published just a 2 or 3 page paper outlining the structure of the DNA molecules with the arrangement of all of the nucleotides. And this sort of set off a chain of rapid discovery about DNA replication. A lot of work began in understanding the processes of transcription and translation and the role of RNA and proteins in those processes.